Hello and welcome to day 2 of MD plus DI's Focus on Fundamentals course, proposing a test strategy to fulfill both the labeling, CMR, and safety evaluation requirements by combining a quantitative, targeted, and a semi-quantitative screening approach. It's sponsored by Nelson Laboratories. My name is Peter Krass and I'll be your moderator today. Before we get started, I have a few quick announcements. First of all, the webinar is designed to be interactive. There's a dock of widgets at the bottom of your screen, and that will help you to learn about today's speakers, download resources, share the webinar via social media, and participate in the question and answer session that we'll have toward the end of the presentation. Secondly, the slides will advance automatically throughout the event. You can also download a copy of the slides, and you can do that with the resources widget. Toward the end of the webinar, I'll ask you to complete a survey, and that's found on the right-hand side of your screen. I hope you'll take a minute to fill out this survey before you leave us today. Your feedback provides us with valuable information on how we can improve events in the future. And finally, if you have any technical issues during this class, please click on the Help widget that's found at the bottom of your screen, or you can type your issue right into the Q&A. Either way, we'll be glad to offer one-on-one assistance. Let's move on to our class. Today, our main speaker is Thor Rollins. He's Senior Director of Advisory Services at Nelson Laboratories. We will also have some additional commentary and help with the Q&A from Dr. Matthew R. Jorgensen. He's a Senior Chemist and Toxicologist, also at Nelson Laboratories. To learn more about our speakers, you can visit the bios, like biographies widget that's at the bottom of your screen. With that, I'm going to hand it over to Thor Rollins. Thor.
Thanks, Peter, and thank you, everyone, for joining us today. We really enjoy these webinars. We actually enjoy doing this in person a little bit more, but because of circumstances that I think we're all very familiar with, these webinars are a great option to be able to talk about potential impacts that are out there in the industry. And today I'm going to be talking a little bit about the labeling of the CMR. And for those of you that maybe are not familiar, hopefully most of you guys are, but the CMR stands for carcinogens, mutagens, and reproductive toxins. And this is mostly coming from the Annex 1, Section 10.4 of the new MDR, the medical device regulations that are coming effective in May. That was pushed back a year because of COVID for Europe. So if you are working with a notified body to have a CE mark in Europe, I'm hoping you're familiar with the MDR and specifically this Section 10.4. Now, just to kind of set the stage, the spirit behind the reason why the CMR was put in was to really help limit the amount of these carcinogens, mutagens, and reproductive toxins. There's lists out there that you can find in the document from getting out in the environment. So these medical devices, most of them will be thrown away at some point, and they want to try to understand how much of these CMRs are going to be out there in the environment. But the way they have put this requirement in the MDR makes it a little bit more interesting for medical devices to be able to fulfill it. So what we've come up with, and a lot of kudos have to go to, her name is Annalise Bertanen over in Leuven, our Leuven office. She's the one that helped come up with this flow chart that we're going to be showing. And it's kind of a proposed strategy to fulfilling both the labeling and also the safety requirements when you're looking at these CMRs. So for us, what I'm going to start doing is building on Dr. Jorgensen's presentation yesterday about analytical chemistry, because it's going to play potentially a very big role in helping us evaluate these carcinogens, mutagens, and reproductive toxins. But to do that, we have to try to understand the difference between a screening technique, which Matt talked about a little bit yesterday, and a targeting technique. So we're going to spend a little bit of time going a little bit more in depth of those two things today. And what this really means is when you get your chemistry back for an extractable leachable, you'll notice that we'll have tentatively identified, most probable, and unknown compounds. And you'll see a splattering of different types of those compounds depending on the device and the library that's being used. And so this plays a role in the CMR approach, and we'll talk about what that means in a second. But to try to help understand why we see these differences and what does it mean when we're doing a targeted versus screening approach, what I like to compare it to something in real life is fishing. So we are based in Utah. It's got some of the best fishing, at least mountain fishing, stream fishing, and so we'd highly recommend you guys come visit us anytime you would like to do some fishing and hiking. I'll take you personally. But what's the big difference between fishing? There's two really kind of basic ways you can fish. One is more of a targeted fishing approach, and the other one's a net fishing approach. And so when you're targeting fishing, right, you're looking for a specific type of fish usually. So you kind of know the lure that these fish like, you know what's the best time of day to go fishing to catch that specific fish, and you probably know a lot about that fish in general. If you're looking for rainbow trout, you know exactly what a rainbow trout looks like when you catch it, you know what it tastes like when you cook them. So you're very familiar with that fish and how to catch it. So it's pretty much in your toolbox. It's something that you've done many times, and you're very confident that you can catch one and know when you have caught one. The net fishing, though, is a little bit different. So when you throw out a net, usually you're not looking for, I mean, maybe you're looking for a specific type of fish, but that net doesn't care. It's going to catch whatever fish is in that area. And the size of the netting is going to determine the size of the fish. So the more closely woven the net is, the smaller the fish you're going to get, but the more bigger gaps there are in that weave, the more fish that can get through. You're going to have fish that you've probably seen before and you're familiar with, but you're probably going to get fish that maybe you haven't seen before, it's new, and you may not know exactly everything there is about that fish. And maybe where you're depending on where you're fishing, some of those fish may not be very good to eat or may even be poisonous. And so when you start looking at this a little bit into a hybrid between fishing and chemistry, so when we look at a targeted approach, so remember this is the targeted fishing approach. So what when you end up doing is you end up looking for that fish. So in the stream or lake, there could be other fish out there. There could be very little minnows or, other types of maybe salmon or other things, but you're looking for that specific fish. And in this case, we're looking for that tuna species A. So when we target that fish, we don't know what else is in that stream necessarily. We're really looking just at that tuna. And so all we'll get is that tuna. And let's just say we're so good of a fisherman that we're able to get all the tuna out of that lake or body of water that we're fishing the stream. And so we can realistically tell you the exact amount of tuna in that body of water if we're that good of fishermen, but we're not able to tell you if there's any sharks or other fish in that body of water because we were just focused on catching that tuna. When you start looking at the screening approach or the net fishing approach, once again, remember that the net size is going to affect if you can catch all the fish or not, right? So if you could throw out just basically a plastic net and you could catch everything, but it's going to be very hard to fish that way. It's going to be very difficult to even physically be able to fish that way. So that's why they have obviously the woven nets, but depending on the gaps in that, you're going to lose some fish of different sizes. And then when you actually open that net and look at the fish, there may be fish that you kind of know the species, or the family. So you might know it's a tuna, but you may not know exactly what kind of tuna it is. So you may be familiar enough with the family of tuna to be able to identify it as a family of tuna, but you may not know the specific species. But you may be able to identify the salmon in there and say, yes, I know that's a salmon. I've seen those before, and I 100% know that that's a salmon. But you're also going to get other fish that you probably don't know. You've never seen before. You don't really know anything about them. So that's the benefit and disadvantages of these types of fishing approaches, is trying to understand, focused on one type of particular fish when you target fish versus that net where you just grab everything in there at certain sizes. The amount of fish in the net, though, is interesting. So if you weigh the net with all that fish in place, and you're trying to estimate the size of those fish in the net, you could say, okay, so I know a minnow weighs this much, and so I could divide that weight by the minnow weight, and that should tell me how much weight each of these fish are. Or I could estimate using the weights of the tuna, right? And depending on which one you pick, that helps you you're estimating the weight of each fish. But obviously, if you choose the minnow versus the tuna as your standard of weight, it's going to drastically change the estimate of each of the fish. So you got to be careful where a targeted fish, you can weigh that fish that you have and know exactly this, because it's always going to be tuna. You know by the standard weight of tuna about how much tuna you have. So how do we apply all this fishing concept with this to the MDRs, the CMRs? So like I mentioned, here is the exact point in the MDR in Annex 1. And the interesting part up above is it talks about devices, right? So that's going to be an interesting interpretation of this standard. Devices or those parts thereof, the materials that they're in, and then it goes down to having to contain anything above 0.1% by weight of a CMR present. So basically, interpreting the legalese here is that in your device, if you have more than 0.1% by weight of a particular CMR, you have to label that device as having that CMR. Now there's so much gaps there in my opinion, because that doesn't, like I said, this is meant for in the environment, devices that are going to be thrown away, how much CMR is going to be put out there, because if I have a very heavy device, the percent of CMR by weight versus a very light device from us as toxicologists, that's a big difference. And so for me, that doesn't really help me understand the impact to the patient at all. What it's really just doing is, once again, kind of screening for potential environmental impact down the road. But needless to say, we have to evaluate the amount of these CMRs in our devices. So we could do a targeted approach, kind of like that targeted screening, and we could look for a very certain CMR. And in this particular example, let's say we have a polycarbonate plastic. And in this device, we know we have polycarbonate. We know that bisphenol A is it's a plasticizer that's likely in polycarbonates. So we could definitely target this CMR and just look for that particular CMR. We would use the GCMS method to evaluate this particular case, and we'd be able to know for sure if the amount of bisphenol A that's leaching from a device from this particular polymer. And we could tell you exactly that's there and how much is there, because this is a targeted approach. So it's like that targeted screening approach. But did we capture all of the possible CMRs in there? Even if it's polycarbonate and we pretty much know that this is the potential CMR in the polycarbonate, there's other sources that could be giving CMR to this device that besides just this plasticizer. You could be looking at the manufacturing process, the shippings, any colorants that may be put into the plastic, the sterilization processing could have an impact potentially. Basically, we have to be able to understand, and did they add anything other on that list of CMRs besides this one plasticizer? So the screening technique helps us kind of brush through. So we may do a targeted technique looking for specific CMRs, but we can't just say that's it, we're done. We've identified the possibility of that 0.1% by weight without doing some kind of screening for anything else that could be there that's unknown. So the target approach is limited for the known amount of targets. You're only going after those particular ones. You're using a selective technique, so we have ICP-MS for elements, IC for anions, and it has to be that validated method for specific compounds. Where the screening technique, There's no predefined target, just like that net fishing. We're not looking for a specific fish, so we screen for them, but we could be missing compounds, right? So our net could be not close enough, and some of these CMRs could be leaching. We could be not knowing what they look like, and so they could be lost with not being identified as a CMR. But we would go through this process very similar to what Matt talked about yesterday, just using some general screening techniques that are found in ISO 10993-18 to try to apply screening for these CMRs. So those are the two techniques you could go through, and honestly, you're probably going to have to use a combination of both of them to really understand the full potential of your CMRs in your device. So the other part of this is you got to remember is that besides just identifying these fish or compounds, we also have to look at that weight, like I talked about with the net. And a screening versus targeted approach gives us different types of quantification amounts. It gives us different values of that weight. And remember, going back to the net, comparing the average weight of each fish by using the weight of a minnow versus a tuna is going to give us a totally different average. And so when you start looking at these types of amounts, it's important to try to understand the values you get from a targeted versus a screening approach when you look at masses or amounts. So when you look at quantification, the screening approach, the net approach, does give you an estimated or semi-quantification method because you're averaging the total amount of fish by some kind of standard. Now, if you just use one internal standard to do that, like a minnow or tuna, you may be off depending on how your fish compares to that minnow or standard. If you use multiple types of different fish, right? So if you have a very small minnow to a large tuna, but then you also have maybe a trout or things in the middle, and you can kind of divide out unknown fish to most resemble those weights, that gives you a more accurate semi-quantification. So the way you have semi-quantification is important to remember. But your target approach will give you full quantification. So we'll know exactly how much is there because it's targeted. Okay, so these are just some of the quick definitions. I put this more in here for your own use. These are definitions that come from the standards. So there's definitions of quantification and identification that helps you understand kind of more scientific-based descriptions of what we just talked about. Okay, so let's actually get into the approach that we would do when we start looking at a CMR. So we've kind of set the stage a little bit with a screening approach versus targeted approach using chemistry. But the problem with that is you got to remember that when we run chemistry, we're only looking for the leachables or extractables, depending on how we set up the device, or these chemistry, coming off of the device. That doesn't tell you the total amount by weight. And that's the biggest gap with you as medical device manufacturers trying to do this yourself, is that unless we can totally dissolve the material and run it through our instruments, we're never going to be able to tell you total content. And that goes back to that body of water. So unless we're incredibly good fishermen and we're able to say we got everything out of this body of water that's a tuna, we're only going to be able to tell you an estimate based on what we see leaching off from the device. So the first step in any CMR evaluation when you're looking to fulfill this for your MDR submissions should be going back to your suppliers. Your suppliers should have this information. They should be able to give you, saying my material doesn't have more than these list of CMRs by percent. I do want to warn you though, having done this now for a couple of years with many of our clients, this isn't a foolproof approach, and you kind of have to base your approach on how much trust you want to put in your suppliers versus how much do you want to find out for yourself. For example, we've had many people who have had different plastics that they claim do not have plasticizers that are on the CMR list, like BPA. And when we do the testing, we'll find these plasticizers. So we go back to the suppliers, and what they've found out is that the tubing may not have it, but one of the connectors that they also manufacture does. And so there's lots of times that, because I think lots of people are new to this type of thing, maybe they're not quite understanding the total weight by weight percentage kind of issue. But the first step should always be trying to get this from your suppliers. So you ask for your suppliers if they have a concentration present of these CMRs in the device. So go to them, ask them if they can tell you on a concentration, not just if it's there, but actual concentration by weight of these CMRs in your device. And if they do, then you want to see if that CMR is greater than 0.1% by weight for your total device. And this will help you understand if it does or not. So if they can tell you that there's a percentage of weight, or maybe they can't tell you there's a percentage at all, maybe they just tell you that, yeah, I think we have BPA, but I'm not sure how much we have in there, then you have to do that full digestion of the material if you can. That's a big if, because not all materials, one, we can degrade, and two, when we do degrade, can then be ran through chemistry. But we want to try to do a full digestion and then do that targeted analysis for that specific CMR that the company, their supplier, claims is in there to get the full quantification. So hopefully they can give you a percentage weight by weight so you don't have to go down that route. And once you do, you go back to make sure that is not above that 0.1% by weight in your CMRs. If it is above 0.1% by weight, that's okay. Honestly, granted, I'm not sure exactly what the market pressures are going to be after this, but for the most part, it doesn't mean you can't label or sell your device in Europe. What it means, you just have to label your device as having that particular CMR, and also the amount, so that it can be later tracked and identified. If it's not above 0.1% weight, so if you do have BPA in your device, but it's not above that 0.1% by weight, then you don't even have to label it. You're fine. So you can just move on from there from a particular labeling standpoint. But this doesn't help. This is one of my biggest pet peeves about this whole concept, is it doesn't actually address anything to the risk by patient. Like I said, if that 0.1% by weight, when you start, and if you attended yesterday's presentation by Dr. Du, from toxicology, we really care about dose to the patient. And this doesn't tell me anything about how much CMR, or these carcinogens, mutagens, or reproductive toxins are actually going to the patient per day. And so even if you have it or you don't, we still have to even if it's above or below 0.1% by weight, we should do some kind of risk evaluation to the patient per ISO 10993-17. Now, so let's say we still want to screen too, even if the information, the concentration is there. We want to screen for any of these possible CMRs just to make sure that we're not having any surprises from, like I said, connectors, or maybe from processing that we may not know it's there. And if you can't get any information from your suppliers, really the only chance you have, the only way you can really try to address this question is by doing this semi-quantitative screening approach. Because, like I said, trying to get this information by digesting or dissolving the material doesn't always work. And so if you can't get this information from suppliers, then that screening approach sometimes is our only chance to screen for possible CMRs. Now, I do want to tell you that the chance of these things being so high in concentration and not coming out, so 0.1% by weight is pretty high. So if they're in there by 0.1%, they are likely to come out. And so we're likely to see them at low levels. So this is how we would set up a chemical characterization, not just for safety risk assessment, but also for the CMRs. So we'd still make it an extract per 1093-18. Once again, if you were attending yesterday's session, you heard Matt kind of talk about the difference between sample preparation between -18 and -12. But we would follow very simple steps in our chemical characterization that's just like 1093-18 for chemical characterization. So we'd extracted the same kind of situations. We make sure there's no physical change in the devices, that the solvents didn't impact the device. And then we'd run them through our normal screening techniques using our VOCs, SVOCs, and non-VOCs, and ICP-MS for metals. And so these are our techniques that we would normally go through. And we give estimated semi-quantitative results of every compound that we would find. So our net would just pick up every finish that we could detect based on that. and using, like I said, elements at ICP-MS giving full quantitative results. So that concept, that's how we do a chemical characterization like Matt reviewed yesterday. So setting up one for the safety risk assessment for CMRs is very similar. So we still make the extract, but this is for the identification, the targeted approach. So instead of doing a screening though, we may just do one technique like this SVOC we have up there, and we do that calibration curve to target BPA. So that's the big difference between the screening and the targeted technique for CMR assessments. Now, if we don't know if a CMR is present, then this is the step process we would recommend. So it's very similar to that 2993-18, but the biggest difference is once we get the estimated semi-quantitative results, we would then gather all those fish together from our net, and we would compare that with the published CMR list. So basically what we're trying to do, think about it this way with the fishing and the net, is we gather all those fish together, we bring over like a guide who knew these really poisonous fish, or maybe the fish that we're not supposed to catch, and he or she would go through our net and be able to show us which fish are on that list. Now remember, these are estimated and semi-quantitative. There may be different identifications, so we may have some that are ticks or tentatively identified as a CMR, but at least it's given us information about what could be coming off our devices that could fall into that CMR list. Then the step 2 would be to go back and actually see if these CMRs that maybe are tentatively identified or maybe even most probable, and then actually get a calibration curve for that particular CMR to be able to confirm that that's there and then give an accurate, not a semi-quantitative, but a more quantitative result for that CMR. So we'd want to take those results and refine them using a second step. Now, this helps you understand the benefits and disadvantages of each of the screening and targeting technique. So when we target something, it is fully quantitative, and that's great. And so we have low uncertainty, but it's obviously just that one compound. So we don't really get a very idea of anything else. Where the screening, we have more estimation, there's more uncertainty, but we're able to see through much more of these compounds. So really what we're trying to do is overlap these, where we get some really good semi-quantitative estimations and identification using a robust response factor technique and a library review to be able to feel confident that we're able to identify and quantify, at least tentatively and semi-quantitatively, these CMRs, and allows us to go back and make a more targeted technique after we're seeing them in our screening technique. So when you start looking at the flow now, going through the information and the process, first you want to see, is there information present? If there is, from the supplier, right? You do that chemical characterization. If there's no information from the supplier available, you do the chemical characterization per -18. Then you evaluate the risk to the patient, which is an important part because then you can start using those results for other types of systemic evaluations like systemic toxicities and carcinogenicities and immunogenicity endpoints. And then you start doing your CMR known list, and then you go into your CMR targeted list if you have them. But any of those compounds, even the CMRs, should then go into a toxicological assessment per -17. Because like I said, for me, I don't really care if it's less than or greater than 0.1% by weight, because that doesn't tell me the risk to the patient, because it doesn't give me a dose to the patient. What I need to figure out is how much of it's coming off per day to the patient, what's the dose, to be able to understand if it's a true risk to the patient going forward. So if that concentration is close to 0.1% weight, though, if it's on that list and it's close, then we try to do that full digestion. And this is the part that I really wish we could do, because then that gives us a total amount in there by weight. But like I said, this is a lot of times not feasible based on the materials and the chemical techniques. But then we can still compare that to the percent by weight of CMR, if it's greater than 0.1 or not, to be able to label those as having a CMR or not. So this is the proposed workflow that we have for CMR. So just thinking about it, the first, going through it just really high level one more time. First, you want to ask for that information from a supplier if they have it in their material. If they're able to give you that information, ask for concentration. Hopefully they'll tell you it's a concentration of total concentration, and then you compare that on the weight of your device, percent by weight, and then if it's above 0.1, you have to label that. If they tell you there's a CMR there, but they can't tell you the concentration, then we try to do a full digestion to be able to evaluate that amount per weight. If we can't do a full digestion, or if they can't give you any information, then we jump to a chemical characterization screening approach per 10993-18, where we're evaluating the risk of the patient, and we're also looking at the results or net of fish compared to the CMR list. If we see CMRs on that, then we go back over to try to do a full digestion to be able to label. Or we also do a toxicological risk assessment to understand the risk of the patient. The only real, I guess, hole in this possibility of workflow is if you do not get information from your supplier and you do the chemistry and you find CMRs there, but we're not able to digest that material or run it on chemistry, then you're really kind of stuck justifying using the screening and targeted approach of leachables or an extractable at this point profile instead of doing a full digestion. And I would propose to really go back to the supplier and say, look, we found this particular CMR in your device. We know it's there. You are now being held responsible as a supplier to identify these kind of material, these compounds. So please let us know the total amount there, because if not, you're kind of stuck using an extractable semi-quant, or even if it's quant, it's still an extractable amount and not the full concentration in the device. So these are the proposed workflows. We've actually done this many times now with clients. We've had a few of those go through notified bodies and have had some really good success. I do have to say that that when you start looking at the submission process, though, we've only had a few of our clients that weren't able to get this information from the suppliers. And so we did have a couple of them that have submitted chemistry targeted extractable results for their CMRs, and we're still waiting to hear back about the conversation with the notified bodies with that approach. Most of our clients have been able to get this information from their suppliers. I do just want to warn one more time, if I was in your situation, I would, even if I got that information from my supplier, I would still want to do some kind of screening for CMRs, just to be sure, because I've seen too many times where the supplier says there's not one there, and we find one. So just be careful there. So I just want to point out that obviously we don't have as much time as we'd like to really get into a lot of the details in screening and chemistry and toxicology and biocompatibility or any of our possible solutions. So we offer a lot of educational events and resources on our website. That's both Nelson Labs and Sterigenics. So Nelson Labs is more around the testing and evaluation of medical devices. Sterigenics is more obviously focused around the actual sterilization side of things. Both of those are very good educational resources where we have webinars and videos that we can help with those. So a big thank you to Matt for helping me with this and co-presenting with me on these presentations. At this time, we are open up for questions. So Peter, I think we'll take it over to you.
Thank you, Thor. Thanks. That was great. We are going to go to Q&A, but before we do, I want to remind everyone about the webinar survey, which I mentioned earlier. Again, it's on the right of the presentation window. If you close the survey, you can reopen the widget. Just click the icon along the bottom of your screen. Again, thank you in advance for filling out the feedback form. Your participation in this survey will help us to serve you better in the future. Okay, let's get into the Q&A. It is not too late. You still have time to ask a question. just type it into the text box that's located to the right of the presentation window. Or you can click the Q&A icon at the bottom of your screen. And let's jump in. We've got lots of questions, which is great. The first one comes from Barry, who asks, is the 0.1% limit based on the weight of the compound or the weight of the device?
That's a great question. So let me go back to the actual verbiage in section 10.4 of the MDR. So it says devices or those parts thereof or those materials used therein. And then it says that it shall only contain the following substances in a concentration that is above 0.1% weight by weight, where justified to section da. So when we've had this discussion with a lot of our clients, we've had clients that have read that and they've interpreted that they could evaluate that 0.1% by weight by either the weight of the device, the weight of the parts, or the weight of the material. And so we've actually seen companies that say, okay, my device has DHP in it, or this material has that plasticizer in it, and it's above 0.1% by weight by that material, but it's a very small piece in my device. So the device's weight is greater, so the amount of DHP is not greater than 0.1% by the device. And so they've decided not to label it. For me, as I interpret this, it says I would take it wherever is the worst case, because obviously as a risk assessor, that's how we look. And so when I start looking at it, I think it tells you that those are the three things you should evaluate: the device, the parts, and the materials. And if any of those have something that's 0.1% by weight and concentration higher, then you should label. So if I had a connector that had DHP and I had like a distal end that had DHP, I would incorporate those together as looking at the device, but then I'd also look at the individual components of the materials and see if any of those are greater than 0.1% by weight. And if they are, any three of those, I would then have to label. So it's not necessarily the concentration of the DHP, it is the concentration of that in any of those three areas is how I interpret it. Matt, do you have anything you want to add to that? That's how we've been, I mean, like I said, it's kind of up to legalese, it's kind of up how you want to interpret it, but for me, that's what the spirit of the law says. What do you think, Matt?
Yeah. So I totally agree. I think that in the MDR itself, it leaves it open to choose one. If I look back on other EU regulations that like REACH that have similar thresholds, in those cases, it sounds like it's per component. So it depends if you're following the letter of the law or trying to read into what historical precedence is. But when we've done these evaluations in-house, we prefer to look at all three. And as you said, 0.1% is kind of like a high. It's kind of like a high limit. So it's not that hard to comply with the most conservative, version of that.
Okay, great. Well, we have our next question. This comes in from Sabiha. Hope I'm pronouncing that correctly. Can you give us some examples of products that are covered by this MDR regulation?
Oh, wow. So this basically any product that is looking to be marketed in the EU. And by the way, this does also do Class 1 devices, even though you're not submitting those to a notified body, you're the notified body kind of in that case. You still want to make sure you're following the principles in MDR because you could be audited, and you want to make sure that those devices are covered. So any medical device that's considered a medical device in Europe that's looking to get marketed there has to follow the new MDR, which means that you have to at least consider this 0.1% by weight requirement.
Okay. Our next question comes from David, who's asking for a clarification. If the risk to the patient is acceptable and the weight is below 0.1%, does that mean there's no requirement to label?
Yes. Yeah, that's great clarification. So here's the funny thing. Even if the risk to the patient is there, but it's still less than 0.1% by weight, you wouldn't necessarily have to label it, but you'd still have to address that to your notified body, right? You'd have to try to justify why this toxicological risk is there. So the labeling really is just that 0.1%. But if you have less than 0.1% by weight of a CMR, and you're able to show from a toxicological risk assessment that that amount is not a toxicological risk, then you do not have to label your device.
Okay. Satish has a question. For compounds where there is no CMR limit, or maybe that's not available, do we consider the TTC limits for toxicological assessment?
You know, Matt, since I could definitely answer this, but since you're the board-certified toxicologist, why don't you take this one?
Yeah. So that's a great question. So what I would say is, just to be totally clear here, that these CMR limits aren't protective of patient safety at all, right? So you can't rely on a CMR limit to argue patient safety in any way. So if we're talking about compliance to this part of the MDR from like a legal perspective, this is where CMRs come into play. If we are concerned with demonstrating patient safety to an EU regulatory body, then it's we won't look at the CMR list. We'll take a look at the chemistry results and compare those to both the TTC and other toxicological information available.
Okay, great. Next question comes from Jennifer, who asks, when you go to a supplier to ask for information, are there particular CMRs you need to ask about? Is there a defined list that we can refer to?
Yeah, there is a list. So you can actually reference that list in the CMR. So that list is constantly updated. So you'd want to make sure you get the most current version. And so you could just send them that list or reference that list when you ask them. The other thing is there are certain materials that have, like, we have a pretty good indication that there's probably one of these CMRs listed present, like cobalt, for example, or nickel even. And then you're also looking at these plasticizers. So these are things that you may specifically call out depending on what your material is, but you can definitely give them that list.
Okay. Next question comes from Johan. It's about metal devices. He asks, does it make sense to include screening for VOCs if you have a metal device where volatile contamination could have reasonably evaporated from the product before it can be detected or before it can get to the patient?
Yeah, so this is a very good question, and Dr. Jorgensen, definitely fill in where you feel is appropriate. But the truth is that metal products tend to have less VOCs than polymers, right? But that doesn't mean that we never see VOCs. And so we end up seeing sometimes VOCs coming from metal products. It could be contaminants, and maybe they're just not evaporating off because they're trapped somewhere under maybe an oil or something like that. So for me, even though you're probably less likely to see a lot of VOCs coming off that could impact the patient, we still want to run it because you can't guarantee that VOCs would not be there.
Great. Thank you. Our next question comes from Thomas, who asks, how do you address CMRs that are present in manufacturing contact materials? Specifically, how do you determine the weight of the CMR substance?
Yeah, so if I understand your question correctly, it's just not the materials that could contain it, but the processing that could leave residual CMRs. So actually, that one's a little bit easier because the hard part is, like I said, digesting that material. When you try to digest something, you try to get total content, or like a surface contamination from a process, sometimes it's easier. Now, if the contamination is in the mix, then that becomes more difficult. But what a lot of our clients end up doing is they take that CMR list and they go through their processing. Now, the whole there is it's not just your processing you're worried about, it's your supplier's processing, their supplier's processing, and sometimes their supplier's processing. And that's why the screening comes into play. But we still have our customers go through their processing, the chemicals they use, and compare a lot of those ingredients to the CMR list, just so that we can maybe do more targeted approach on those compounds if we know what we're looking for beforehand. So, Matt, did you have anything you wanted to add to that one?
No, I think that covers it. I guess I would just say, like, if you know that it's just an external contacting material, so it's going to be only on the surface, then, you might be able to argue that, since it's not part of the material, it's more like a surface residual, that it's more about residual manufacturing materials. So that's one thing. And the other thing is that it's really unlikely that any surface contaminant could be present above 0.1%. So normally through a kind of like a cursory review, we can conclude that those surface residuals wouldn't be a concern. If it ends up in the mix, like Thor said, then it's definitely part of this.
Great. Okay, our next question comes from Alexander. Here's his question. What is the process requirement prior to labeling 0.1%? What investigation is required to show due diligence that there was an effort to replace the hazardous material?
Yeah, so that's a great question, and actually that is something that we do recommend when we are able to identify either a CMR that's above 0.1% by weight or a CMR that's obviously a toxicological concern. Is the first thing we always try to have our clients do is replace that material. Now, a lot of times that's easier said than done, right? So like even a plasticizer, trying to find a different plasticizer, even though they may have replacement plasticizers, not always can we use the plasticizer we want, or the changing it may be very hard. So we do have clients that have decided just to label it and not go through the whole changing a material technique at their point. But you should evaluate the pain and the cost and the time of replacing the material versus the labeling. And for me, that I know that has to go with in business, like being able to sell the device if there's pressures about having a DHP on your label. For me, it's more about safety. And so for me, the real decision comes if I have enough CMRs in there to cause a cancer concern or a toxicological concern, then I would definitely, obviously, want to replace that material. If you're safe, then it goes more into a business selling kind of discussion about the pre-labeling and removing of that material.
Okay, good. The question now comes from Chad. Any insight into how cobalt in SS is being handled, as cobalt was recently added to the CMR list?
Yeah, Chad, this is a great question, and I'm going to throw out just not cobalt, but I'm going to throw out nickel, right? So there are a couple metals on the CMR list that are commonly found in medical device materials like stainless steel and cobalt chrome and others. Honestly, we have not really got good direction from anybody in Europe, any notified bodies or anybody that we know on the European Commission on how they're exactly going to handle these obviously well-used, known, safe materials. All I've been able to hear from these people is that, remember, this isn't necessarily a safety evaluation. It's an evaluation for the, these out in the ecology, right, out in letting these out in the environment. And so they do want to know if they're releasing a ton of cobalt or a lot of nickel. And so they still going to probably require labeling, but obviously the amount of nickel and cobalt, we have levels that we know these, and most of the time the leaching amount of these is very much lower, so we can justify safety, but the labeling will probably have to be there.
Okay. Our next question comes from Eva, who asks, could you briefly explain how and where to search for CRMs in the databases that are available for this?
Yeah, Matt, I'll let you take this one since you're the one that does it for us.
Yeah, I'm happy to. So Ava, we monitor these lists ourselves, right? Because we do these screens all the time, and so the legal code inside the MDR. So if you go to section 10.4 of the MDR and look there, then there's a code, and it refers to the CLP. And the CMRs are a subset of the CLP list. It's the CLP Annex 6. And so if you Google search for the CLP Annex 6, what you can find is the status of that list in PDF format when it was published. And then you can also find on ECHA updates to the CLP Annex 6 list. And so I would go to the one that's on ECHA, and you can download that in Excel format. And then the CLP Annex 6 contains a lot more chemicals than just CMRs. It has lots of other hazardous chemicals on there as well. So what you want to do is filter that list in Excel to just select, carcinogens, mutagens, and reproductive toxicants. And then the endocrine disruptor part of 10.4 of the MDR, I think that one actually refers to the endocrine disruptor subset of the REACH list. And so basically, if you have both of those lists and you can filter them, then that's I guess you would say the legally binding CMR and endocrine disruptor list. And then they do get updated from time to time.
Okay, thanks, Matt. Next question comes from Monica, wondering which chemical characterization techniques does Nelson Labs use in-house?
Yeah, we use pretty much all of them from Dash 18. So we have, the GCMSs, the HPLCs, and ICPs. We have, so looking for volatiles, semi-volatiles, non-volatiles, metals. We also have, some of the screening ones like FTIR. So a lot of those the common techniques that we can do in-house. Matt, am I missing any that you can think of?
I guess the only thing I would add is, so Headspace GCMS, regular GCMS, but then in addition to HPLC and LCMS, we have high-resolution accurate mass. So that's really kind of a more specialized technique that's required. So we have, a nice LC Orbitrap to screen in LC, which I think is an important part of that.
Yeah.
Okay, good. Our next question comes from Kenneth, and he's asking about differences between Europe and the U.S. Here's the question. MSDS made for Europe are supposed to indicate CMRs when present over 0.1%. Do U.S. SDSs made according to OSHA have the same type of requirement?
So here's the truth, and I'm going to say this before Matt gets on his soapbox. So even the requirements, though. So honestly, SDSs or MSDSs, we have a hard time really trusting those, even the requirements, because they're not peer reviewed. They're not really done necessarily by the right kind of people or using the right techniques. So it's not saying all of them are bad, because obviously some of them are very good and you can trust. But just to throw a blanket out there, it's hard for us to even trust the ones that are required. But for FDA or for US, no, not all of them even will have. They'll have a hazardous section material and they'll have on there, but they can put unknown. I mean, Matt, do you have anything you want to say there? It's not really a good source as far as questioning from U.S. suppliers.
Yeah, I mean, I'm glad you jumped in, because I was about to get up on my soapbox.
I've been working with you long enough to know that you'd be on the soapbox.
So the only thing I would add, and I guess you did kind of already say it, but I have talked to like industrial hygienists and folks that work at OSHA about any particular requirements for like the quality or peer review of these SDS sheets, and there basically aren't there. And I've also talked to lots of people at chemical supplying companies, and I asked to speak to the person who writes their SDS. And I mean, no offense to any people or also pizza delivery people, but I mean, I wouldn't trust these SDS writers sometimes to make me a pizza. You know what I mean? So there's not a lot of requirement there to make sure that the quality of these SDS sheets are where they need to be to make any determination on patient safety. Let's just put it that way.
Okay. Our next question comes from Ed. It's about digestion. He says, you mentioned full digestion if you can't get full chemical composition or CMR composition from the supplier. Does Nelson Labs do full digestion for EU MDR analysis?
Well, we try. So like I mentioned in the presentation, that is something that's in the step. That's one of the things that we want to do, because when you look at the spirit of the MDR, it's not about the leachable profile, even though you can argue that if it's in there 0.1%, you would see it's probably leaching. But the truth of it is you want to know the total concentration. So the only way to really know a total concentration, if you don't get that from the supplier, is to digest it. And so we do offer that, and we do try, but there are materials you can't do, and there's a lot of materials you could probably digest, but then to be able to get something that you could then analyze on an instrument, you either have to dilute it so much that it becomes hard to hit certain sensitivity levels. So it is a step that we try to do, but it's one, honestly, that not always we can do.
Great. Our next question comes from Tuba, who asks, can we use the same approach you've presented here for the ED substances?
Yeah. I mean, I don't see why not. I'm not as familiar with those list of substances, but it should, I'm somewhat familiar with them. And so I can't think of a reason why you shouldn't. Matt, do you think of any reason you couldn't? I think it would work.
No, yeah. So the methods that we have for screening are really broad. And when we reviewed both the CMR list and the endocrine disrupting list, our professional opinion was that, okay, there's some endocrine disruptors on there that are more like biologically derived, substances that we probably wouldn't detect, but we know aren't on there anyways. And then the ones that are, artificial, those ones we would expect to capture in our screening. So we feel confident that, we're covering all the ones that could be there.
And the only thing I would add to that is anytime you're looking at an endocrine disruptor compounds, it's probably a good first step just to know what those compounds are and understand if it's even possible to have those in your device. Honestly, a lot of those are probably not, like Matt just said, possible to be in there. And so we really want to look. Matt has a presentation where he talks about why are we looking for moon dust if we know that moon dust is not going to be in the device. Some of these endocrine disruptors are very similar to that. So we want to be focused, but like Matt said, we should be able to catch them all.
Great. Looks like we have time for one more. This comes from Satish, who asks, do we need to label a device even after doing the risk-benefit analysis?
Yes, unfortunately. So we've actually had this discussion with notified bodies and a person in the European Commission who helped write the MDR. So remember, the whole point of this, with the CMRs, isn't patient safety necessarily. It's more about environmental safety and impact. And so even if you're able to show that it's not a toxicological risk to the patient, but it's still above 0.1% weight, you still have to label it because they're wanting to be able to evaluate these or limit the amount of these CMRs out in the waste in the environment. I'm not quite sure how they're going to do that, but I guess if it's labeled, maybe they'll be able to know more, be aware of it. But to kind of answer your question, yes, you still have to label it even if you show it's safe.
Okay, thank you. Looks like we're just about at the top of the hour. There were a couple questions we didn't have a chance to get to. We will share the questions with Thor and Matt, and hopefully they'll be able to get back to you by email. Gentlemen, thank you. Any closing comments before we go?
Not for me. I just want to say thank you for everybody who took time today or yesterday and today, and then just remind you tomorrow we have another great presentation. We love talking about these things, and if anybody has a specific question, they can always just reach out to Matt or I. We'd love to help with anything that we can.
Excellent. I'd like to let everyone know that within the next 24 hours, you'll receive a personalized follow-up email. This will contain details and a link to today's presentation on demand. If you have friends or colleagues and peers who couldn't join us today, tell them about this. They can listen to the event on demand. This webinar is copyright 2020 by Informa Markets. The presentation materials are owned by or copyrighted by Nelson Laboratories, and the individual speakers are solely responsible for their content and opinions. I'm Peter Krass. I'd like to thank you for attending today's course, and please be sure to join us tomorrow. Thank you.